Today I'd like to tell you about the research we do in my lab studying gastrointestinal motility and the regulation of motility. Before I do that though, I'd like to tell you a short story. When I first came to Brockport, I had the opportunity to talk with my father, some longer conversations because he wasn't healthy. And he told me about his troubles that he had with diverticulitis. That's a disease that's an inflammatory disease, and it's a gastrointestinal motility disorder. The most important thing in my father's life was work, and he never missed a day of work. It turned out, with this disease, he'd miss several days in a row. I was pretty surprised by that. The second thing he told me is he couldn't go see NASCAR races. And it turns out he's a social person, and he really liked going to the car races at the neighborhood bar with his friends. So for this disease to keep him home, stuck in his apartment by himself, a really social person who had placed a high value on work, it showed me how important gastrointestinal motility is and how much we're lacking in treatment for these disorders. I had studied GI motility for 15 years. I was a patch clamper, and I studied really small electrical currents that flow across the membrane of a smooth muscle cell, and it's really straightforward to understand how those cells contract when the currents flow. However, I never really thought closely about the entire system and the influence that these diseases would have on people. So when I got to Brockport, I really thought long and hard about what I wanted to do, and I wanted to have a system where I could study GI motility without perturbing it. Even though all the processes that control motility are really complicated, and that's why we take them out of the animal to study them in isolation, and then we relate how it would work in the whole system, I wanted to see the whole system. Of course, there are problems with that and challenges, but I thought the benefits really outweighed the problems. So I decided to use the zebrafish model, and you can see why in this slide. This big picture is a movie, and it's sped up. What you can see on the bottom is the gastrointestinal tract, sometimes called the digestive tube, and you can see these small contractions. I took this video in my friend's lab at the Mayo Clinic. I went there because they had some tools that I didn't have, and Shu Chalet, another friend there, studied zebrafish. So he provided me with some 7-day-old animals, and we fed them a little bit of blue dye. We just put it in the water, and you can see here that the GI tract is easily visualized in this A, B, C, D small panel. We could also really visualize the contractions that we want to study so much and that we want to understand how they're regulated. So this was a decision to go forward. The next thing we did was we looked at a mutant animal, and Stacy Hess did this work. She was a student in my lab at the time, a really great student. And what you can see on the right, these long rectangles, is just the gut of the zebrafish. The top panel is a normal zebrafish, and the bottom panel is a mutant. You can see the gut is bigger, and that's just like what we can see in the mouse. We were so excited when we saw this because it really gave us an indication that the zebrafish was likely to be a good model system to study GI motility. Scott Ledden, another student in the lab, took these images, and what he was able to do was to really visualize one cell type, the interstitial cell of Cajal, in the GI tract of the zebrafish. This work had never been done, and it showed us that the important elements that control motility, and that's smooth muscle cells, enteric neurons, other people knew that those existed, and this pacemaker cell, the interstitial cell of Cajal, that's what we brought to the table, that existed in the zebrafish as well. Right in the outer muscular layer of that digestive tube, you can see that in that pink figure in B really well. So we have a model that we can really visualize, and the next thing we needed was a way to quantify GI motility. And that's what this movie shows. What you can see in this looping movie is the contraction propagating down the GI tract and these small ripples in that image beneath it. And Grant Hennig, another good friend from the University of Nevada, Reno, helped me with this. He used this technique for something else, but we applied it to the zebrafish, and it allowed us for the first time to really quantify movement in the GI tract. So this was really powerful. We showed that ICC and all the elements to control GI motility are in the gut, and we also developed a way to measure it. So we've been moving forward and we're studying the role of a new gene called anoctamin-1. And what you can see in this rotating image is the network of pacemaker cells or interstitial cells of Gehal in the wall of the GI tract. So we use a laser scanning confocal microscope, and we took this image right here at Brockport in my lab. I want to move forward and tell you what's coming next. So we've been able to show that the model is really useful. We can see the whole GI tract in this animal. And now the question is, what can we do with this model that we can't do with other models to help us learn more? Well, we know ICC are present, but how do they develop? That's an unknown question, and we think we have a really good opportunity to study this in this animal that's transparent. The second thing we want to know is, can they be repaired after injury? We know people with motility disorders, like my dad, have problems with ICC. And the question is, can they be fixed? That's the ultimate treatment. And to know if they can be fixed, we have to know what the molecular pathways are. So I want to conclude by thanking all these people who helped me with the work, the students at Brockport, my colleagues, and of course the college for supporting me. There's the National Institute of Health that funds this work. And then there's my good friends, Gianrico Ferrugia, Simon Gibbons, and Shu Chalet at the Mayo Clinic, and Grant Hennig at the University of Nevada, Reno.